Nervous tissue is made up of two principal types of cells, neuroglial cells and neurons. Neuroglial cells are like the nerve glue of the central nervous system. They play a big role in holding it together. Some neuroglial cells function to support and nourish neurons. Some surround a synapse and play a significant role in communication between neurons. Glia cells are the only nervous system cell that can undergo mitosis. Neurons do not. So they are the only type of nervous system cell that can metastasize or develop cancer. We're pretty much born with all the total number of neurons we're going to get in our lifetime. However, we will learn about neuroplasticity, the process by which neurons can grow in size and increase their number of synapses. The neuron's primary job is to receive and transmit signals. They are the facilitators of communication in the body. There are several types of neuroglial cells. We're going to talk about 6. 4 in the central nervous system, called astrocytes, epidermal cells, oligodendrocytes, and microglia cells. The 2 in the peripheral nervous system are called Schwann cells and satellite cells. Astrocytes. As you can see in the diagram, astrocytes are star-shaped, and that's how they got their name. They are the most abundant type of glia cell found in the CNS. The processes that extend off the body of an astrocyte wrap around capillaries and nearby neurons, effectively anchoring the neuron to the capillary. This position allows astrocytes to mediate nutrient and waste exchange between the blood and the neuron and support the blood-brain barrier. Astrocytes and the blood-brain barrier. Astrocytes play an important role in forming and maintaining the blood-brain barrier. In the previous slide, I described processes that arise off the astrocytes that wrap around the capillaries. Well, in addition to providing structural support, astrocytes secrete chemical messengers called paracrines that stimulate the capillaries to form and maintain tight junctions between their endothelial cells. So the layers that make up the blood-brain barrier going from inside the vessel to out are endothelial cells held together by tight junctions maintained by astrocytes, a basement membrane, and the podocyte portion of an astrocyte process. Astrocytes and synapses. Astrocytes also surround synapses as demonstrated in the diagram. They have a significant role in maintaining the concentration of potassium and neurotransmitters in the interstitial fluid within the synaptic cleft, which is the space between two communicating neurons. Remember, we covered potassium leaky channels in the cell membrane? Well, that includes the neurolemma of a neuron, and if too much potassium is concentrated in the synaptic cleft, the receiving neuron might not be able to respond to a stimulus. Hopefully these terms I'm using are not foreign to you, but if they are, we will cover them in the anatomy of synapses later on in this course. Astrocytes are extremely important in the recycling of neurotransmitters, glutamate and GABA. We'll cover that process when we get to the topic of neurotransmitters. Astrocytes in the immune system. Astrocytes have a significant role in the repair of CNS tissue post-insult. Insult to neurons in the central nervous system can be caused by infection, trauma, ischemia, stroke, autoimmune response, and other neurodegenerative diseases. In the early stages after an insult, astrocytes not only activate inflammation, but they also form barriers that limit immune cell migration and therefore the spread of potentially neurotoxic inflammation, meaning they control the flow of the immune response. Additionally, astrocytes secrete neurotropins called nerve growth factors, which will stimulate an injured neuron to begin its repair process. Microglia cells comprise 10 to 15% of brain cells. It's not as much as astrocytes, but it's still a large portion. These cells that arise from hematopoietic stem cells and not nervous tissue play a role in the CNS immune system. In fact, they are the first and primary line of defense in the central nervous system. I often refer to them as the cleanup crew because a big part of their job is to remove debris resulting from tissue insult. I covered the types of tissue insults in the previous slide, but I'd like to add multiple sclerosis and lack of use to this list. We'll cover lack of use with neuroplasticity, but basically, if you don't use a neuronal synapse, the microglia cells will remove it and take it to the trash. We'll see microglia activity in neuropathophysiology when we cover Parkinson's disease and Alzheimer's disease. Epidermal cells and oligodendrocytes. There isn't much to say about these two glia cells. Epidermal cells line the ventricles in the brain and the spinal canal within the spinal cord. Their main role is to sweep cerebrospinal fluid along with their cilia. Oligodendrocytes form myelin, a whitish lipid around CNS neuron fibers. This myelin helps to increase the speed of nerve signals in the central nervous system. Satellite cells of the PNS. Now we've moved on to the glia cells of the peripheral nervous system. The main function of satellite cells is to support and nourish neurons by mediating nutrient and waste exchange and to regulate the local chemical environment. Their function in the peripheral nervous system is very similar to astrocytes' function in the CNS. Schwann cells of the PNS. Schwann cells are the most important glia cell of the peripheral nervous system. Similar to oligodendrocytes of the CNS, Schwann cells also produce the lipoprotein called myelin that forms concentric sheets around long axons of neurons. And as in the CNS, this myelin increases the speed of nerve transmission down the axon. But that's not the only function of Schwann cells. Just like microglia cells in the CNS, Schwann cells remove cellular debris. And like astrocytes, Schwann cells play a significant role in the growth and repair of injured neurons. Myelin protein 0. The myelin produced by Schwann cells wraps around a neuron's axon up to 100 times. Now, in order to prevent the myelin sheath from unwinding, it needs some type of substance to hold the concentric rings together. This substance is called myelin protein 0, also known as P0 or MPZ. Myelin protein 0 is an adhesion protein molecule that functions to hold Schwann cells together. Problems with this adhesive protein is associated with approximately 29 neuropathies. It is most commonly associated with a condition called Charcot-Marie Tooth disease, which is a genetic condition that we will cover in neuropathophysiology. Anatomy of a neuron. All neurons have three basic parts: the cell body, dendrites, and an axon. The cell body is also known as the soma or perikaryon. The nucleus is located within the cell body and contains the neuron's DNA. Dendrites are processes that project off the neuron cell body. Little buds arise off the dendrites called dendritic spines. Most neuronal synapses in the brain occur between the axon terminal of a sending neuron and the dendritic spine of the receiving neuron. We will learn later on that neuroplasticity involves the growth, development, and pruning of these dendritic spines. One large, long process arises off of the cell body called the axon. A cone-shaped thickening of the soma, called the axon hillock, gives rise to the initial segment of an axon. Many axons are covered with myelin. The end of the axon branches into multiple processes called telodendria. These terminal ends of the telodendria form swellings called axon terminal. The axon terminal has several synonyms that I'll mention later on. Neurotransmitters are stored within the vesicles inside these axon terminals. Neuron organelles. Neurons have organelles just like every other cell in the body. Some of the organelles we will cover are smooth and rough endoplasmic reticulum, Nicel bodies, which are unique to neurons, Golgi apparatus or Golgi body as I learned it, lysosomes, mitochondria, neurotubules, and neurofilaments. Endoplasmic reticulum, or ER. Endoplasmic reticulum has a series of interconnected tubular membranes that is continuous with the nuclear envelope. There are two types, smooth ER and rough ER. The smooth ER is called smooth because it does not have any ribosomes. Rough ER is called rough because it is covered with ribosomes. Smooth ER produces phospholipids to maintain the plasma membrane and stores intracellular calcium. The rough ER produces proteins, transport proteins for the plasma membrane and neurotransmitters, and export proteins. Nisobodies are unique to neurons. They are cytoplasmic masses composed of flattened stacks of rough ER surrounded by free ribosomes. These nisobodies also undergo protein synthesis, producing transmembrane proteins, neuropeptides, enzymes, and so on. These nisobodies are only found in the soma and dendrites, and they have a significant role in neuroplasticity. Nisobodies are not found in the axon. Golgi, lysosomes, and mitochondria. The Golgi apparatus, or Golgi body, is very similar in structure to smooth ER. In fact, it is also composed of smooth-walled cisternae. The Golgi receive proteins produced by the ribosomes, then package these proteins in vesicles and ships them out into the cytoplasm. For this course, we will focus on the large molecule neurotransmitter packaged by the Golgi. Lysosomes are tiny structures that contain acidic digestive enzymes called hydrolase. This hydrolase is strong enough to break down worn-out organelles, cellular waste, viruses, bacteria, micro and macro molecules. If a lysosome were to rupture inside the cell, it would digest the cell itself. And of course, the mitochondria are for energy production. The neuron cytoskeleton is formed from neurofilaments and neurotubules. These microfilaments and microtubules have a haphazard arrangement in the soma, but are linear in axons and dendrites. The main function of neurotubules is to form the framework for intracellular transport. Dendrites make up approximately 90% of the surface area of a neuron. Their main job is to receive information from other neurons. Dendrites can carry information to and from the soma, but most of the time they carry information towards the soma. The more dendrites a cell has, the more input it can receive, and the more input a neuron receives, the more important that neuron is. The proximal region of a dendrite is composed primarily of nisobodies, Golgi, neurotubules, and neurofilaments. This anatomical feature is important when we cover how neuroplasticity functions. Dendritic spines are knobby projections on the cerebral and cerebellar dendrites. These knobby projections are the location of most axonal dendritic synapses. The knobby projections, or buds, permits neurons to have thousands of synapses at one time. Dendritic spines have a significant role in neuroplasticity. Axons are long projections that rise from the axon hillock, which is a thickened cone-shaped area of the soma. Each neuron will have one axon and only one. The first portion of the axon is about 30 to 40 micrometers long and is called the initial segment. The initial segment is also known as the trigger zone in motor neurons because it is the region of the neuron where most nerve impulses are generated. Myelinated axons in the peripheral nervous system have unmyelinated spaces approximately 1 micrometer between the swan cells, and these are called the nodes of Ranvier. Telodendrons. I will basically repeat the same description I gave on slide 12 about these structures, but according to Google, you have to hear something 7 times before you remember it. And if Google says it's so, then you know it's true. Anyway, with my description, telodendron are long, slender projections at the terminal end of an axon. An axon can have just one telodendron or several telodendria. The characteristics of telodendria are that they are slender, vary in length, and are always unmyelinated. When we get to the topic of nerve physiology, knowing the anatomical structure of telodendria will help you understand why the speed of a nerve impulse slows down significantly once it reaches the telodendria. The terminal end of telodendria forms synaptic bulbs, also known as synaptic knobs, also known as terminal boutons, also known as axon terminals, which is my favorite term. and all the other synonyms for this structure. It is at the axon terminal that will form a synapse with another structure. In this course, we will focus on neuron-to-neuron synapses and neuron-to-muscle synapses. Axon terminals, as I mentioned previously, are the terminal end of an axon, and more specifically, the terminal end of the axon's telodendron. Axon terminals produce small molecule neurotransmitters and store them in vesicles. The axon terminal releases both small and large molecule neurotransmitters via exocytosis through the presynaptic membrane. The presynaptic membrane is the most distal surface of the axon terminal, and it is the surface that faces the synaptic cleft. The synaptic cleft is the interstitial fluid-filled space between the axon terminal and the structure with which the axon is communicating. Axon collaterals are extensions or shoots off the axon that branch off the main or parent axon. These collaterals will have the exact same properties as the parent axon. Collaterals will have the same diameter, and if the parent axon is myelinated, they'll be myelinated. If the parent axon is unmyelinated, then they'll be unmyelinated. In myelinated peripheral neurons, the collaterals will only arise off of the nodes of Ranvier spaces. Axon conduction. Rate of impulse through the axon is determined by three characteristics: the axon's diameter, length, and whether or not it's myelinated. The larger the diameter of an axon, the greater the rate of the nerve impulse conduction, and vice versa. The smaller the diameter of an axon, the slower the rate of nerve conduction. The length of an axon also determines the rate of the nerve impulse conduction. The longer the axon, the longer it takes for signals to travel back and forth. It takes a lot longer for an L5 nerve signal to reach your big toe than it takes for a nerve signal from C8 to reach your pinky finger. As mentioned previously, the lipoprotein myelin will increase the speed or rate of a nerve impulse that travels through the axon.